अस्सलाम वालेकुम स्टूडेंट्स। तो आज हम डिस्कस करते हैं एपोप्टोसिस को। तो एपोप्टोसिस इज अ caspase dependent program cell death and caspase is a protease enzyme and this process is controlled by multiple genes include the pro-apoptotic gene and the anti-apoptotic genes. अब हम इसको डिफाइन करते हैं the series of the morphological and the physiological changes that occur during the program well regulated cell death is known as apoptosis. अब हम बात करते हैं about the characteristic of the apoptosis. So the apoptosis involves single cell or smaller group of the cell. In apoptosis, there is a shrinkage of the cell occur due to the destruction of the structural protein, whereas in necrosis, the cellular swelling occur. Similarly, in apoptosis, the cell membrane remain intact and hence there will be no release of the cellular contents occur, due to which there will be no inflammation occur in the apoptosis and the cellular contents that are packaged in the form of the apoptotic bodies. And the apoptosis is a type of the suicidal cell death that occur in the benefit of the human body, and it is a active, well-regulated process. And apoptosis is important for the development of the hemostasis and the elimination of the pathogens and the tumor cells from the body. And the apoptosis may be physiological or the pathological, whereas the necrosis is only pathological process. Physiologically include the endometrial shedding of the cell that occur during the menstrual cycle or during oogenesis. Around about 15 to 20 follicles are selected, but all the follicles undergo apoptosis and only one follicle remain and become mature. इसी तरह there is apoptosis of the virally infected and the new plastic cell by the T killer cell. There is apoptosis of the self-reactive T or the B cell during the normal hematopoiesis. And the pathological examples include the DNA damage, misfolding of the protein, and the viral infection of the hepatocytes that lead to the council main apoptotic bodies, or the organ transplant rejection, or retinitis pigmentosa that involve apoptosis or degeneration of the rods and cones, and the glandular atrophy that occur due to the luminal or the ductal obstruction. And the excessive apoptosis occur in the neurodegenerative disease like Alzheimer's disease and the viral infection, and the reduced apoptosis occur in the autoimmune disorder. अब हम बात करते हैं about the genes that are involved in the process of the apoptosis. So they include the pro-apoptotic genes. These genes induces the process of the apoptosis, and they include the BAX gene, the BAC gene, P53 gene, which is an important pro-apoptotic gene, and the corticosteroids hormone. And the anti-apoptotic genes, these genes inhibit the process of the apoptosis, and they include the BCL2 gene, BCLXL gene, and the MCL1 gene. And the third one is the sensor family. And this family include genes which maintain the balance of the apoptosis, and they involve BID gene, BIM gene, Puma gene, Noxa gene, and a normal cell, the BCL2 gene expresses and they produces protein which are present in the mitochondrial membrane, and they inhibit the process of apoptosis by preventing the release of the cytochrome C from the mitochondria. But when there is the absence of the growth factors or hormones occur, then the BCL2 family is replaced by the pro-apoptotic genes like BEX and the BEX gene, which are activated, and they causes the increased permeability of the mitochondrial membrane to cytochrome C, and the cytochrome C is released, which leads to the activation of the apoptotic pathway. अब हम बात करते हैं about the pathways of the apoptosis. तो उसमें सबसे पहले intrinsic pathway. It is also known as the mitochondrial pathway of apoptosis. And the stimulus by intrinsic pathway is the decrease in the growth factor or the misfolded protein or the DNA damage. और जब हमारे साथ growth factors कम हो जाते हैं तो there will be a decrease in the BCL2 activation, which is an anti-apoptotic gene, and this gene is replaced by the BEX and the BEC family of the gene, which are the pro-apoptotic gene, and they induces the process of apoptosis by increasing the mitochondrial permeability to the cytochrome C, and there will be the release of the secondary mitochondrial activator caspases, which blocks the activity of the inhibitor of the apoptotic proteins. And when the inhibitor of apoptotic proteins are blocked, the process of apoptosis will occur. When the cytochrome C is released, it causes activation of the apoptotic activating factor 1, which leads to the activation of the caspase 9. And the caspase 9 is a protease enzyme, and it is an initiator K space. Whenever K space 9 is activated, it leads to the activation of the K space 3, 6, and 7, which are the executioner K spaces. And when these K spaces are activated, they lead to the activation of the protease and the endonuclease enzyme. And the protease causes breakdown of the cytoskeleton, whereas endonuclease causes breakdown of the DNA and the chromatin condensation. And the cellular contents that are formed, that are packaged in the form of the apoptotic bodies. And these apoptotic bodies then undergoes phagocytosis by the macrophages. तो इंट्रिंसिक पाथवे में हमारे साथ ग्रोथ फैक्टर्स कम हो जाते हैं और देयर विल बी डिक्रीज इन द एक्टिवेशन ऑफ द एंटी एपोप्टोटिक जीन वेयर इज द प्रो एपोप्टोटिक जीन्स आर एक्टिवेटेड लाइक बैक्स एंड द बैक फैमिली और हमारे साथ माइटोकॉन्ड्रियल मेम्ब्रेन की परमेबिलिटी बढ़ जाती है साइटोक्रोम रिलीज सी रिलीज होता है जो कि एपोप्टोटिक एक्टिवेटेड फैक्टर 1 को एक्टिवेट करता है जो कि हमारे साथ एक्टिवेट करेगा केस्पेस 9 को केस्पेस 9 हमारे साथ केस्पेस 3 6 को एक्टिवेट करता है जो कि फिर हमारे साथ पादा प्रोटीएसेस और एंडोन्यूक्लियस को एक्टिवेट करता है एंड दे कॉज ब्रेक डाउन ऑफ द सेलुलर कंटेंट्स and these cellular contents are packaged in the form of the apoptotic bodies which then undergoes phagocytosis. अब दूसरा हमारे साथ है एक्सट्रिंसिक पाथवे तो इसको हम डेथ के पाथवे भी कहते हैं और द रिसेप्टर लिगेंड पाथवे भी कहते हैं और इस पाथवे में हमारे साथ जो पेस लिगेंड है ट्यूमर नेक्रोसिस फैक्टर अल्फा दे बाइंड टू द पेस receptors and these receptor belong to the CD5 CD95 receptor family and these receptors are the death receptor and when it is activated they causes activation of the FAD which are the pace associated dead domain and they leads to the conversion of the pro-caspase 8 and to the caspase 8 or the caspase 10. The caspase 8 is present in the animal whereas caspase 10 is present in the humans and whenever these are activated they causes the further activation of the executioner caspases like caspase 3, 6 and the 7. Whenever these enzymes are activated, they lead to activation of proteases, which causes the breakdown of cytoskeleton protein, and the endonucleases, which causes the breakdown of the DNA. And the cellular content form, they are packaged in the form of the apoptotic bodies. And these apoptotic bodies then attract the macrophages, which causes apoptosis, and there will be no inflammation occur. अब हम बात करते हैं about the caspases. तो caspases are the cysteine containing protease enzyme and these caspases may be initiator caspase or the executioner caspase. जो initiator caspases है, they include the caspase 9 which is present in the intrinsic pathway and the caspase 8 or the caspase 10 they are present in the extrinsic pathway and the caspase 8 is found in the animal whereas the caspase 10 is found in the human and the executioner caspases involve caspase 3, 6 and the 7. अब तीसरा हमारे साथ है साइटोटॉक्सिक टी सेल मीडिएटेड pathway. तो cytotoxic cell को हम T killer cell भी कहते हैं and when these cells are activated they releases purpurins and the grain enzymes. The purpurins make a pores in the cell whereas the grain enzyme enter through these pores and they activate the caspases which leads to the apoptosis and the example of this pathway include the killing of the virally infected cell. अब हमारे साथ एपोप्टोसिस में जो एपोप्टोटिक बॉडीज बन कहते हैं these apoptotic bodies attract macrophages and undergoes apoptosis. एपोप्टोसिस में हमारे साथ फ्लिपिंग का कंसेप्ट है नॉर्मली जो सेल मेम्ब्रेन है इसमें जो फॉस्फोलिपिड्स होते हैं नॉर्मल सेल में दे आर प्रेजेंट ऑन द इनसाइड or the inner side of the cell membrane but during apoptosis these molecules undergo flipping over the cell membrane and they comes toward the outer side of the cell membrane and these compounds include phosphatidylserine or the thrombospondin molecules and these compounds attract macrophages and undergoes phagocytosis and these compounds can also bind to the annexin 5 stain which is used in the estimation of the process of the apoptosis. अब आ जाते हैं about the diagnosis of the apoptosis. So it can be determined by the chromatin condensation by the hemotoxylin or the fuel gene stain, estimation of the annexin 5 stain, which binds to the phosphatidylserine or the thrombospondin molecule. And similarly, the apoptosis also shows the step ladder pattern on the gel electrophoresis. And similarly, the DNA breakdown can be also measured by the tunnel technique. And the last one include the estimation of the cytochrome C level. अब बात करते हैं about the pyoptosis. तो pyoptosis occur in the cells that are infected by the microbe and this process the activation of caspase 1 occur in the caspase 10. Caspase 1 activation further lead to activation of interleukin 1 which induces inflammation and leads to the symptoms of the fever whereas the caspase 10 activation causes cell death. The morphological changes in pyoptosis include the cellular swelling and the inflammation. आखिर में आता है हमारे साथ necroapoptosis. So they include both the necrosis and the apoptosis and it is the caspase independent pathway because there is no involvement of the caspase protease enzyme occur. In this pathway, the tumor necrosis factor bind with the tumor necrosis factor receptor, which further lead to activation of RIP1 and the RIP3 genes, which are the receptor-associated kinases, and hence there will be a formation of reactive oxygen species in the cell occur. Similarly, the ATP synthesis also decreases if the lysosomal membrane are damaged, the cell membrane rupture, and the contents are released from the cell, which induces the inflammatory process. And the example of necroapoptosis include the pancreatitis, reperfusion injury, and the Parkinson disease.